Hello, everyone. In this chapter of the cardiovascular system, we are going to address specifically the blood. As we dive into the blood, a few questions I would like you to keep in mind. One, it's what type of tissue is the blood? What are the different components of the blood, both cellular and non cellular? What is the function of these different components that are found as being part of the blood? What do you need to know before blood donation? What are antigens? What are antibodies? We are going to dive into that concept. How are the ABO blood types determined? How do you go about that? What types are compatible for blood transfusion in terms of blood types, right? And finally, we are going to take a look at the rhesus factor. Why is the rhesus factor important when it comes to pregnancy? So these are some of the questions I would like you to keep in mind as you are diving, as we are diving in this chapter. Now, first, what is the function or what are the functions of the blood? The blood is going to be very important to transport nutrients, oxygen, waste, including carbon dioxide and byproduct of chemical reactions and hormones. So the blood is going to be used as transportation system, right? Two, it's going to play a very important role in the defense in the body against infectious particles, including pathogens, right? Finally, the blood is going to play a regulatory function. And that regulatory function is going to involve many sub-functions, including playing a role in the control of body temperature, the water-salt balance, and the body pH. All of this falls, you understand this by now, in homeostasis. So the blood plays a very important role in maintaining a relatively internal stable environment. Now, what is the composition of the blood? I want you to remember that the blood is made up of two major components. You have the cellular components and the non-cellular components, right? Now, overall, know that the blood is a type of fluid connective tissue. That fluid connective tissue is going to be made up of a bunch of water, proteins, carbohydrates, amino acids, and cells. The cells fall in the group that we call formed elements. These formed elements are going to have basically three major types of cellular components. The red blood cells, also called erythrocytes, I want you to remember that fancy name. The white blood cells, also called leukocytes, I also want you to remember that fancy name, the leukocyte, and the platelets, also called thrombocytes. Please remember for each of the items the two names. Red blood cells, erythrocytes, white blood cells, leukocytes, platelets, thrombocytes. It's very important to remember these names because if you go, for instance, in a hospital and say they want to analyze your blood, they can talk about erythrocytes rather than talking about red blood cells, right? You have to know what they are talking about. Likewise, when they talk about white blood cell, they can very well say oh, the leukocyte count is low or the leukocyte count is high. Are you going to know what they're talking about? So, very important to know both names. Now, the formed elements are the cellular component of the blood. How about the non-cellular component of the blood? The non-cellular component of the blood is called plasma. Plasma is going to be made up of bunch of water, so 91% of the plasma is water, while 9% of the plasma is salts or ions. You can think of calcium, potassium, sodium, magnesium, you name it, right? But also, in addition to salt, you are going to have a lot of organic molecules, including sugars, amino acids, right? Proteins, okay? Now, in addition to the first components we talked about here, we have plasma proteins. The plasma proteins are going to be the most abundant as organic molecules in the plasma. Okay, the plasma proteins are going to be the most abundant. In other words, you are not going to have more sugar than proteins in the plasma, for instance, right? That's what you need to understand. And now, diving into the plasma proteins, what are the most abundant plasma proteins? By far the most abundant, it's going to be a protein that we call albumin. Albumin is going to be very important in terms of abundance in the plasma and is going to play a very important role in the osmotic pressure, as well as helping in the transport of nutrients. In other words, albumin is going to attach to some nutrients for the sake of carrying them, right? Then the next most abundant protein in the plasma is going to be the globulins. The globulins play a very important role in the transport of chemicals in through the body, right? And among the globulins, you are going to have some globulins that are not necessarily involved in the transport, but that are doing other jobs, such as the protection. Now, these globulins that are involved in protection are called immunoglobulins. 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 That's the fancy name for antibodies. Antibodies. All right? And then the third major protein that is found in the plasma, it's going to be the fibrinogen. The fibrinogen plays a very important role in blood clotting. The fibrinogen are these proteins that when they connect to each other like this, they form long threads. These threads eventually are going to form a network, and that network, it's what is going to prevent the blood from leaving blood vessels when there is a breach, right? The blood clot, it's basically you're going to see a lot of network of fibrins like this, forming structures like this, so that the red blood cells do not go through the small spaces here. So the red blood cell would be like big like this, so the red blood cell cannot go through, right? All right. So looking at the cells that make up the blood, these cells, they all originate in bone marrow. Okay? So say we have a long bone right here. The red bone marrow is going to be located at the extremity of long bones. This is where you are going to have stem cells that, based on the type of signal they are going to receive, are going to differentiate. Differentiate into red blood cells, white blood cells, and platelets. Okay? So before a red blood cell becomes a red blood cell, it is first of all a baby red blood cell that we call erythroblast. That erythroblast is going to mature and become a red blood cell, which job is to transport oxygen and carbon dioxide. Now, for some stem cells that are going to become white blood cells, right? You're going to have different kind of baby white blood cells. These baby leukocytes are going to be lymphoblasts, monoblasts, and myeloblasts. The lymphoblasts are going to mature into lymphocytes. These lymphocytes are very important and instrumental in specific immunity, while the monoblasts are going to differentiate into monocytes that, when activated, become macrophages. These macrophages are responsible for engulfing foreign particles, including bacteria and viruses. Then the myeloblasts are going to differentiate into different kind of cells, including neutrophils, which job is to engulf foreign particles, including bacterial pathogens, right, viruses. The eosinophils that are going to play a very important role in fighting worm infection and also in allergy. And finally, the basophils are going to be very instrumental in inflammatory response, right? They're going to release histamine and play a role in inflammatory response. Now, the megakaryoblast are going to differentiate into megakaryocytes. These megakaryocytes are going to split into many fragments that we call platelets or thrombocytes, which are instrumental in blood clotting. Now, let's take a look at red blood cells. The red blood cells are very unique in the sense that they do not have a nucleus, because red blood cells are the only cells in the body that do not have a nucleus. Well, guess what? These cells are not going to be able to multiply. Once they are red blood cells, they cannot multiply. All right? And also, in addition to the fact that they are lacking a nucleus, they are also going to have only few organelles. And again, this whole structural organization is not compatible with reproduction, right? So that's one very important piece of information to remember about this red blood cells. Now, the other thing that is important to know is that the red blood cell, they have a particular shape. They are biconcave. They are biconcave, so they are kind of flat, right? And because they are kind of flat, they can basically navigate very easily in blood capillaries that are the smallest blood vessels you can imagine, right? Each red blood cell is going to contain a particular set of globulins that are called hemoglobin. That hemoglobin is responsible for carrying oxygen and carbon dioxide. There are about 280 million molecules of hemoglobin in a red blood cell or erythrocytes, and each molecule of hemoglobin is going to carry 3 molecules of oxygen. Now, between you and I'm not going to ask you how many molecules of hemoglobins are in a red blood cell. That is not important for me. But what is important is for you to know that red blood cells contain hemoglobin molecules, and hemoglobin molecules are the one that are catching oxygen to carry that oxygen to its final destination. Now, let's take a look at the structure of hemoglobin. Now, you notice that the hemoglobin molecule that is compatible for carrying oxygen, it's not one single molecule of hemoglobin. So this is one molecule of hemoglobin. You have another molecule of hemoglobin, that's 2. You have a third molecule of hemoglobin, that's 3, and 4. So 4 molecules of hemoglobins are complexed with what we call a hem group right here and iron to actually be functional. Okay, so this complex it's in a quaternary structure. A quaternary structure of a protein for the hemoglobin to be functional. Look at the shape of red blood cells here. They are biconcave, so you have a depression on this side, and if you flip the cell, you are going to notice that on the other side, you're also going to see a similar depression, right? That shape allows these red blood cells to be able to actually circulate without problem in the blood capillaries. So now, let's focus on carbon dioxide and how carbon dioxide is transported in the blood. Now remember, the red blood cells, they carry oxygen and carbon dioxide. Now, what is the form in which carbon dioxide is found in the blood? There are 3 forms of carbon dioxide in the blood. The most abundant form of carbon dioxide in the blood, it's in the form of bicarbonate. 68%, about 68% of carbon dioxide is present in the blood in the form of bicarbonate. How so? Basically, you have carbon dioxide that combines with water. You know that water is abundant in our body, right? That combination of carbon dioxide with water is going to produce an intermediate metabolite that is called carbonic acid. That carbonic acid is very damaging to the cell. Now, to avoid that, carbonic acid is going to be broken down into hydrogen ion and bicarbonate ion, right? That bicarbonate ion is the most abundant form of carbon dioxide in the blood. Then you have another form of carbon dioxide in the blood that is actually attached to hemoglobin. And remember, hemoglobin doesn't only attach to oxygen, it also attaches to carbon dioxide. In which case, that hemoglobin that is attached to carbon dioxide is called carbaminohemoglobin. That carbaminohemoglobin in the blood represent about 25% of all the carbon dioxide in the blood. And then finally, you're going to have a very small fraction of carbon dioxide that is present in the blood in forms of gas. That is in terms of 7% of carbon dioxide that is present in the plasma. That is the very reason why whenever we grow human cells in a lab, we can never grow human cells in a lab without carbon dioxide. We always have to supply about 7 to 5% of carbon dioxide to keep the cells happy and multiply. And this is obviously in addition to oxygen, right? Because obviously our cells cannot grow without oxygen. You need oxygen. All right. So, question is, how do we produce red blood cells? Well, we produce red blood cells in conditions when we run low in oxygen. Whenever our blood runs low in oxygen, there are some receptors that are going to send, first of all, feel that there is a low concentration of oxygen in the blood. Then they are going to send the information in the kidneys that are going to now respond to that information. As a response to that information, the kidneys are going to release a hormone called erythrocytes. Erythropoietin. Erythropoietin. Okay? Now, I made a mistake earlier. The sensors are going to sense low level of blood. The information travels in the brain. There we go. The information travels in the brain, and the brain is going to send an information in the kidney to tell the kidney that, wait a minute, you need to release erythropoietin hormone. That erythropoietin is going to travel in the blood and target the red bone marrow. In the red bone marrow, the stem cells there are going to differentiate and become red blood cells. Those red blood cells are now going to go in circulation and cause a capture of oxygen in the lungs, and from there the amount of oxygen level in the blood is going to go back to normal, right? So erythropoietin is the hormone that is responsible for the differentiation of stem cells in the red bone marrow to turn into red blood cells. Okay? So it's very important to remember. Now, the lifespan of red blood cell, it's about 120 days. In other words, if you were to label all your red blood cells today, 120 days later, you are not going to find one single of those red blood cells that you labeled today. You are going to find brand new red blood cells, right? Remember, the red blood cells cannot multiply, right? I want you to remember the role of erythropoietin, EPO, in production of red blood cell. And EPO is released in response to low levels of oxygens. Remember that. Okay? The old red blood cells or erythrocytes are going to be destroyed in the liver and in the spleen. These are the two organs that serves as cemetery for old red blood cells. Now, knowing all this information, specifically that erythropoietin is responsible for increasing the amount of red blood cells in our blood, some athletes are using this information to cheat, right? They do blood doping. Basically, they are going to use erythropoietin, take erythropoietin to increase the amount of red blood cells. And by increasing the amount of red blood cells, they are increasing the chance that they are going to always have a good amount of oxygen in their body, and therefore they are going to be able to increase their athletic performance. They are going to be able to resist fatigue, right? Because of the amount of oxygen and red blood cells they have. So typically what they are going to do, it's many months before an athletic event, they're going to be injected with EPO or erythropoietin. When they inject that, then they are going to produce large amount of red blood cells, and now they are going to be able to outperform other athletes that did not do that. Now, understand that this practice does not go without consequences, because when our body, upon notice that there is low amount of oxygen in the blood, the kidney releases erythropoietin and red blood cells are produced. This is a system that is controlled by our own body. Our own body knows how much erythropoietin to release for a proportionate amount of red blood cells that need to be produced. However, in the case of blood doping, when an athlete is being injected with EPO, well, we decide how much to inject. A physician decides how much to inject. That doesn't take into account the physiological situation of the athlete. As a consequence of that, we can actually and most likely going to inject more than what the physiological need is. As a consequence of that, the athlete will produce so much red blood cells that the blood end up becoming very thick. And by becoming very thick, this is causing the heart to overwork to the point that the heart can give up. And this is when we see some athletes experiencing sudden death, right? When they have a heart attack, because the heart has to work so hard that it gives up altogether. All right, what are some disorder of red blood cells? Some disorder are, for instance, anemia. Anemia, it's a condition when you have low amount of red blood cells or low amount of hemoglobin, right? That is going to cause an individual to actually experience fatigue, right? Not enough energy, because red blood cells equate oxygen, and oxygen equates high amount of ATP. And with low amount of ATP, you don't have enough energy, right? Another condition that we can mention is a sickle cell anemia. The sickle cell anemia, it's a genetic disease that typically occurs among African American. There is a similar disease that is called thalassemia that occurs in Caucasian, but let's go back to sickle cell disease anemia. Sickle cell anemia, it's a disease of the red blood cell where cells, rather than being biconcave, so like this, cells are sickle shaped, like this. And these cells that are sickle shaped, do not negotiate very well the capillaries so that they can go on and block some blood vessels. By blocking these blood vessels, this can induce all kind of complication in a sickle cell patient, right? Now, in addition to that, the sickle cell are going to be prone to rupture so that the amount of normal red blood cells in the body is going to be low, which is going to necessarily lead to anemia. That's why we call the condition sickle cell anemia. And then you have another disease that is called the hemolytic disease of the newborn. This is a condition that we are going to cover extensively at the very end of this chapter. This is a a situation of incompatibility between the blood of an unborn fetus and the blood of mom. We are going to address that when the right time comes toward the very end of this chapter. Now, how about the white blood cells? The white blood cells, there are many kind of them, right? The white blood cells are characterized by the fact that they are involved in protecting you against infections. And they are also involved in allergic reactions. They originate in the red bone marrow. That's where they originate, right? They are going to all have a nucleus, which means they are going to be able to reproduce, right? Now, the signal that is responsible for the differentiation of the stem cells in the red bone marrow into white blood cells, this factor is basically the colony stimulating factor, or CSF. The colony stimulating factor of CSF is the one when the stem cells in the red bone marrow receive them, the stem cells undergo differentiation into white blood cells. Okay? The white blood cells can be found in tissues stored or in circulation in the blood. In the tissues, for instance, the connective tissue, you can find white blood cells in there fighting off infections. Underneath of our skin, you're going to find white blood cells in there. In our lungs, you're going to find white blood cells in there. The white blood cells, their job is to fighting infections. Okay? Fighting infections, they are part of your immune system. Please, this is a very important detail to remember. Those white blood cells, they can live days. Some of them can live months, if not years, or some of them can be for your entire life in your body, right? Maybe you've noticed that some vaccines, when we receive them, they can last many, many years and not have to need a boost. While other vaccines, we have to take them on a regular basis, right? Why? Because some cells that are responsible for protection can last very long, while others may not last very long. All right. So what are the different type of white blood cells? You have roughly two categories of white blood cells. We are going to have white blood cells that have granules. So they're going to have teeny tiny granules in the cells. These are granular white blood cells, or granular leukocyte. These granular leukocytes are going to include neutrophils, eosinophils, and basophils. And then we do have leukocytes that are non-granular, so they don't have granules, right? And these granules, actually, you can only see them once you stain the cells. Until you stain the cells, you are not going to see those granules, right? So these granules right here develop, or you can see them when you are using a red dye, while this granule here, you can see them when you are using a blue dye, right? The agranular leukocytes are going to include the lymphocytes and the monocytes. Now, the good thing about this slide is that this slide also gives you the function of all the different white blood cells. For instance, the neutrophils, their job is going to be to engulf, to phagocytize pathogens, right? They engulf foreign particles, they destroy them so that you are protected. The eosinophils, they are going to be very important at destroying worms, okay, and being very useful in reducing inflammation. The granules they carry contain some enzymes which job is going to be to digest the pathogens, to break down the pathogens and to reduce the inflammation. Then the basophils. The basophils plays a very important role in inflammatory response, right? And that inflammatory response can occur because of an injury or maybe in absence of injury. In any case, they are involved in inflammatory response. The lymphocyte, on the other hand, are responsible for specific immunity. And there we're going to have two kinds of cells, the B lymphocytes and the T lymphocytes. All right? The B lymphocytes are responsible for producing antibodies, while the T lymphocytes are going to provide more of a cellular response, specific cellular response. But let's close that parenthesis. Finally, the monocytes. The monocytes are going to differentiate into macrophages, and there are different kind of macrophages, right? There are different kind of macrophages. But let me just say that the macrophages, their job is to engulf pathogen, to destroy them, and protect you that way. All right, so among the white blood cells, we are going to have the granular white blood cells, neutrophils, eosinophils, basophils, and then we're going to have a granular leukocytes, lymphocytes, and monocytes. All right, so at this point, we're going to go one by one and see if there are some aspects of each white blood cell that I missed. And then we're going to tackle the defects in white blood cells. All right, the neutrophils. The neutrophils are by far the most abundant white blood cell that you are going to find in the body. They represent about 50 to 70% of all the white blood cells. Their nucleus is going to be particularly shaped because it's going to have multiple lobes, right? They are going to be essential for phagocytosis, okay? Engulf and destroy pathogens. Engulf and destroy pathogens. The eosinophils, they are in small amount in the body, okay? They are going to have a bilobed nucleus, and they're going to have a bunch of granules to attack parasitic infections, including worm infections, and also being involved in allergic reaction. Basophils, very much involved in allergic reaction because of their release of histamines. Their nucleus is kind of like U-shaped, but As far as the nucleus is concerned and the shape of the nucleus, don't worry too much about it. This is not an anatomy and physiology class. I'm not going to ask you to characterize the nucleus, right? Rather, I'm going to focus on the function of the white blood cells. Again, I'm going to focus on the function of the white blood cell. I expect you to master the function of the white blood cells. All right, the lymphocytes. The lymphocytes are the second most abundant cells out of all the white blood cells. So they represent about 25 to 35% of all the white blood cells. They are going to have a large nucleus, and that large nucleus speaks to the high level of activity in terms of gene expression in the cells. Some lymphocytes are going to develop into B cells. Others are going to develop into T cells. We are going to see, when we look at the immune system, how these two cells are different and how they are protecting you against infections. All right, the monocytes are relatively uncommon, but these are going to be the largest of all the white blood cells. So when you look at a microscope slide of the blood, and you see blood cells, including red blood cells and white blood cell, the largest cells are going to be the monocytes or macrophages. Okay? So these cells are going to differentiate into macrophages, right? This is the form compatible with engulfing foreign particles. Now, not all monocytes differentiate into macrophages. Some actually differentiate into what we call dendrocytes. 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 But don't worry about it. I'm not going to bug you with it at this point of the course, right? So the job of the macrophages is to engulf, to phagocytize foreign particles. Now, blood cells, as they circulate in the blood capillaries, there is a period of time when these blood cells will have to leave the blood capillaries to go in the tissues where protection is happening, right? They have to squeeze between epithelial cells to find themselves in the tissues here, right? And this process, we call this diapedesis. Diapedesis. That's when white blood cells are squeezing out of blood capillaries to end up in the tissues where they are going to fight off infections. Now, what are some diseases associated with white blood cells? Well, one is, for instance, the severe combined immunodeficiency disease, or SCID. This disease is an inherited disease where there is a lack of some enzymes in the white blood cells, and that lack of enzyme is going to basically going to cause you to not fight off infections very well. Another disorder can be leukemia. Leukemia, it's basically a case of blood cancer, uncontrolled cell division of white blood cells, right? And that is a very bad situation and almost a fatal situation. Another one we want to mention for this course is the infectious mononucleosis. Infectious mononucleosis is also known as the kissing disease because that disease is actually transmitted between people when they kiss. Okay? It's basically caused by a virus, the Epstein-Barr virus. that Epstein-Barr virus is going to infect the lymphocytes, and as the lymphocyte die off, you're going to experience fatigue, sore throat, and swelling of lymph nodes. These are some examples of disorders associated with white blood cells. How about the platelets? The platelets, they come from the red bone marrow, where some blood cells called megakaryoblast become megakaryocytes. These megakaryocytes are the ones that become fragmented. All right? So these fragments make up the platelets or thrombocytes. These thrombocytes, their job is going to be to do blood clotting. Right? Now, the thrombocytes do not work only by themselves to do blood clotting. They work with other clotting factors, including proteins called thrombin and fibrinogen. Together, they are going to be able to do blood clotting. Without each other, blood clotting is not going to happen. Now, let's see what happens during blood clotting. When you have a breach in your skin and you start bleeding, you are bleeding because there was a breach in a blood vessel. So you have a puncture in a blood vessel right here. That puncture quickly is going to be closed by platelets. However, platelets by themselves cannot hold together against each other. You need some other factors to come to actually cause the platelets to stick against each other and for the blood to not go through anymore, so you stop bleeding. So very specifically, what is going to happen is that there is a prothrombin activator that is actually a protein, it's an enzyme that is going to come and activate prothrombin. That prothrombin, in presence of calcium, is going to the prothrombin activator, in presence of calcium, is going to cause prothrombin to become active. And at that point, when prothrombin is active, we call it thrombin. And now, thrombin active, in presence of calcium, is going to cause fibrinogen to become fibrin. And fibrin, it's nothing but fibrinogen activated and attached like this to form long threads. And you're going to have a network of threads, right? Forming things like this. And you're going to see some lattices around, so you can have things like this with little holes right here through which blood cells cannot go, right? So you see the blood cells are so large, and the network of fibrins such that the blood cell cannot go through, the red blood cell cannot go through. And this is how blood clotting occurs, and this is how you stop bleeding, right? Because this red blood cell cannot go across through, you are not going to keep bleeding. But what is interesting is that you stop bleeding, but you still see some fluid coming out. That fluid is kind of yellowish. That's the plasma, right? Remember that the blood is made up of two parts. You have the cellular component and the non-cellular component, the plasma. So in blood clotting, the cellular component is what is going to be trapped so that it cannot leave the blood capillaries or the blood vessels anymore. right? While the non-cellular component, the plasma, is still going to be able to go through. All right. So what are some disorders that are associated with platelets? One disorder that is common is thrombocytopenia. Thrombocytopenia is a disorder where the platelets are too little or they break down very easily. Okay? You are going to be told, for instance, prior to surgery, they're going to quantify the amount of platelets. If the amount of platelet is low, they're not going to perform a surgery. Why? Because you run the risk of bleeding to death. That's not a good news, right? But thrombocytopenia, it's a condition that one can be born with. Okay? Then we have thromboembolism. Thromboembolism is when a clot forms and breaks off from the site where that clot was formed to go plug another blood vessel. We talked about, thromboembolism, right? Thrombus, it's a plaque. Embolism, it's the move of that plaque from one place to another. Okay? Then we have hemophilia. Hemophilia, it's a genetic disorder that, when born with, cause someone to not be able to clot their blood because there is lack of some clotting factors. Now, in terms of blood donation, what do you need to know? Now, it is very important to know that blood donation is safe, right? It's done on a regular basis, and it's a sterile procedure. So you should not be afraid of going and saying, I'm volunteering to do blood donation. Typically, when you go donate the blood, you are going to give about 2 cups worth of blood fluid, right? A pint, basically. You are going to be able to renew some part of the blood you've donated. For instance, the plasma is going to be renewed within a short period of time after you do your blood donation, but red blood cells are going to take a little longer because your kidney has to release erythropoietin, and that erythropoietin has to go in the red bone marrow to cause stem cells to differentiate into red blood cells. And also things to expect, it's maybe you may feel dizzy. This is not always the case. It can happen. You may feel dizzy. So it's good after giving your blood to sit down, eat a snack, and drink some water. These are some recommendations. Now, other things you need to know is that your blood is going to be tested for a number of things, including syphilis, which is a sexually transmitted disease, and the presence of HIV antibodies to see if you were at one time infected with HIV, right? And also hepatitis. Now, upon control of your blood, if, for instance, they notice that, well, there is signs of syphilis, HIV, or hepatitis, you are going to be notified so that you can take further steps for treatment, right? By giving your blood, you can save bunch of lives. You can save bunch of lives. So some of the things you should know, it's that if within the past 12 months, you've experienced some infections, including hepatitis or malaria, or have been treated for syphilis or gonorrhea which are both sexually transmitted diseases, you should not actually offer to do a blood donation. Okay? If you are at risk of having HIV, for instance, if you had a blood transfusion yourself and that blood transfusion came and it was told that blood transfusion actually contained, possibly HIV, then you cannot. If you had sexual intercourses with someone who actually has HIV, you cannot, right? Okay. And obviously, if you have AIDS, so that's the clinical stage of HIV, obviously, you cannot give blood. Now, that we've talked about donating blood, how do we characterize the blood types? Well, to characterize blood types, there are two major things you need to know. One, it's that all cells have cell surface markers, including your red blood cells. So your red blood cells have on their surface some molecules that are characteristics to them, right? Those molecules that are characteristic to your blood cells are going to be called antigens. Now, understand that the molecules that are on the surface of your red blood cells may not be the same molecules that are on the surface of your friend red blood cell or your brother or your sister red blood cell, right? So these molecules are called antigens. What are antigens? Antigens are foreign substances that are capable to stimulate your immune response. In other words, say you breathe something, and that something contains harmful particles such as bacteria. These bacteria are foreign particles to your body. As these bacteria come in your body now is going to develop an immune response, and they're going to produce antibodies. Okay? They're going to produce antibodies to the antigen. And in fact, let me tell you that antigen stand for antibody generator. So the question you need to ask at all time is that any foreign particle you come across with, is it an antibody generator? If the answer is yes, then that particle is an antigen. With that being said, we have in one hand antigens, and on the other hand, the antibodies. The antibodies are these proteins that your body produces in response to a foreign particle that came in your body, right? So antibodies and antigens are the ones that are used to characterize blood type. And this is very important to know before blood transfusion, okay, which is a procedure that is used to save lives, right? Okay? So we notice that based on antigen and antibodies, there are 4 types of blood types. We have blood type A, blood type B, blood type AB, and blood type O. But what is the difference? The difference is that blood type A people are going to have on their red blood cells surface markers that we are going to call A. Blood type B people are going to have, this is not a really good red blood cell, I'm sorry. There we go. This is much better. They're going to have on their surface a different kind of surface marker that we are going to call B, right? So here we have A, here we have B. Individuals with AB are going to have both A and B on the surface of their red blood cells, while individuals that are O are going to have nothing. As a consequence of that, you understand that an individual who is A is going to have antigen A. Therefore, they are not going to produce antibodies against A because they are going to harm themselves, right? They can only produce antibody against B because B, it's a foreign particle. Likewise, B individuals are going to produce antibodies against A because A, it's a foreign particle to them. How about individuals who are AB? Individuals who are AB are not going to produce antibodies against A or against B because A and B are part of themselves, right? And finally, individuals that are O are going to produce antibodies against A and against B because A and B are foreign to them. But now, here is a question that is very important and that I want you to remember. When are these individuals producing antibodies? Well, answer, it's that they can only produce antibodies against a foreign particle when they come in contact with that foreign particle. In as much as they don't come in contact with that foreign particle, they will not produce an antibody against that factor. In other words, if you are blood type A, now, and you never came across blood type B through an accident or anything, you will not have antibodies against B. Okay, you will not. You can only have antibodies against B if by accident you receive blood from someone who is B while you are A, right? That's just an example. All right, so here I'm showing that you have type A here, okay? That's the surface marker in individuals who are A. They're going to produce antibodies Bs. Individuals who are B, they're going to produce antibodies against A, and so on and so forth. You already know the story. Now, blood types are going to be named based on the factor you have on the surface of your cells. If you have the surface marker A, you are going to be blood type A. If you have the surface marker B, you are going to be type B. If you are AB, you have surface marker A and B. If you have nothing, you are O. Right? So I already mentioned all of this, so that's good. Now, here you have a way of basically telling what type of blood type someone is, right? Type A, you're going to have antibodies against B. Type B, you're going to have antibodies against A. Type AB, you're going to have antibodies, type AB, you will not have antibodies against A or against B. You will not. But type O, you will have antibodies against B and against A, right? And basically, this causes us to characterize the different blood type. So When you want to determine a blood type before blood transfusion for compatibility, first consider the antigens found on the donor surface. Second, consider the antibodies that are found or that could be found in the recipient. And then see what the potential outcome is going to be. If the antibodies in the recipient can recognize antigen in the donor blood, this is going to cause a reaction that we call agglutination or clumping. That agglutination of clumping tells you that you should not do blood transfusion at that point. Okay? You should not do blood transfusion. And that blood transfusion will lead to necessarily rejection, right? So no agglutination is a good news. Agglutination is a bad news, right? No agglutination is going to tell you actually that the antibodies are not attaching to the surface markers. Agglutination tells you that antibodies are attaching to the blood surface, right? Okay, agglutination, no transfusion, don't do that. No agglutination, yes, there is transfusion. That can happen, okay? All right. So now, let me test you on your understanding of your blood type and blood donation. First question, can a person with blood type O accept blood? Can a person with blood type O accept blood type A without agglutination occurring? Why or why not? Think about that. Second question, why can people with AB blood type accept more blood types than people with type O, A, or B? That's the second question. The third question is which blood type is able to be used most often as a donor blood type? Why? Think about these questions. Now, before I conclude this chapter, I want you to remember that AB type, a person AB is a universal receiver. A person AB can receive from AB, can receive from A, can receive from B, can receive from O. A person O is a universal donor. He can donate to O, to A, to B, to AB. Okay? So I often say that people who like to receive are selfish. They don't like giving. AB receives from everyone. They only give to themselves. They not give to anyone else. Not to O, not to A, not to AB, not to B, right? And I also say people who like to donate hardly receive. That's the O, right? Donates to everyone, doesn't receive. All right, how about rhesus factor? The rhesus factor, it's another surface marker on the surface of red blood cells. Some of us have that factor on the surface of our cells, some of us don't. Those of us who have the Rh factor are said to be rhesus positive. Those of us who do not have the rhesus factor on the surface of the red blood cell, they are said to be rhesus negative, right? So the rhesus positive and negative occurs both in the population, but this becomes a big deal when it comes to pregnancy. Okay? It becomes a big deal when it comes to pregnancy. Now, if someone has a rhesus factor, so he's rhesus positive, that person obviously is not going to produce antibodies against the rhesus factor. However, if an individual is rhesus negative, that individual is going to produce antibodies against the rhesus factor, obviously when exposed to the rhesus factor, right? So now, let's assume that you have a mom who is rhesus negative, who is married to a man who is himself rhesus positive. Now, because dad is rhesus positive, their future baby, the fetus, is necessarily going to be rhesus positive. Okay? Because the fetus is rhesus positive, that is now an open window for what we call the hemolytic disease of the newborn, right? That hemolytic disease of the newborn can lead to systematic destruction of the fetus red blood cells, or a decrease in the amount of the fetus red blood cells, so that there is a retardation in the development of the fetus, or even spontaneous miscarriage, right? But when does this happen? Now, here in the first pregnancy, let me kind of step back before I talk about the first pregnancy. Let me first of all say that between mom and the fetus, there is what we call placenta barrier. That placenta barrier prevents a movement of the fetus red blood cell to mom's circulatory system. There is no exchange of blood cells between mom and the fetus during pregnancy. So if a mom is rhesus negative and dad is rhesus positive, the baby is born with rhesus positive blood. Now, throughout the first pregnancy, mom was never aware of the difference between her rhesus and the baby rhesus status. Only at birth, this is when the umbilical cord is broken, right? There is bleeding, right? This is when the baby's blood is going to ever come in contact with mommy's blood. Now mom is going to develop antibodies against the rhesus factor here present. Now, these antibodies are going to end up building a memory in the protection system of mom, so that the next pregnancy, actually what is going to happen is that now mom has antibodies. These antibodies are going to be able to cross the placenta barrier and to go in the second baby. And now the fetus here, the blood of the fetus is going to be systematically attacked by the antibodies that mom is releasing. These antibodies are going to cause an agglutination of the fetus red blood cells, so that these red blood cells are going to clump together, and this is going to be a problem for the development of the fetus. That's why A lady who is rhesus negative, during the first pregnancy, the pregnancy goes well, the baby is born, the baby is healthy, and everything is fine. But to have a second pregnancy to be developed throughout and a newborn is born healthy, while that newborn is rhesus positive, the probability is very slim, right? And this is what we call the hemolytic disease of the newborn, right? But knowing this, what can we do to prevent an attack of the fetus blood by mom? What can we do? Typically, when a mom is rhesus negative, after giving birth the first time, she's going to be given antibodies. Now, listen carefully. She's going to be given antibodies against RH within 72 hours of giving birth. What does this do? This does two things. One, it's that the antibodies in circulation are going to go against all the baby blood that went in mom. That's one. So mom is going to capture the baby blood without ever being aware of the presence of that blood. Two, the lack of awareness is going to cause mom to never develop herself antibodies against that blood. Therefore, she doesn't have a memory of that rhesus positive ever, which is actually preparing the body to receive another pregnancy with rhesus positive without even knowing it, right? So mom will never produce antibodies attacking the blood of the fetus, right? And now, when a mom is pregnant again, basically, this is actually when mom is pregnant again, some mom will receive another treatment, right? So that she does not produce antibodies against the rhesus factor. Okay? So that's basically the hemolytic disease of the newborn and how to prevent the hemolytic disease of the newborn. Final slide of this presentation, it's going to be on how the circulatory system works in close relationship with all other systems, including the digestive system, the urinary system, the muscular system, the nervous system, endocrine system, respiratory system, lymphatic system, the skeletal system. Now, between you and I, because of the amount of material, I'm not going to bug you as to how the circulatory system plays a role in homeostasis and how they work with other systems here. But I do highly encourage you to go through the slide and to see how your circulatory system works closely with other systems. For instance, if you consider the cardiac system, the cardiovascular system rather, with the urinary system, how do they work together? So the blood vessels are going to transport fluids in the kidney, and there the blood is going to be filtrated, right? Which is going to actually play a very important role in modulating water-salt balance, that one, modulating the pH, that two, modulating blood pressure, that's three, right? So the cardiovascular system has to work together with the urinary system for these three different aspects of your homeostasis, right? You can do the same thing for instance, the skeletal system with the storage of calcium ions, right? The production of blood cells, including red blood cells and white blood cells, because you know the skeletal system, it's where the red bone marrow is going to be located, right? Okay, so thank you for your attention, and let's close this chapter now.